Biocompatibility. In simple terms, biocompatibility means a medical device can touch your body and still play nice. It does its job without causing harm. No toxic effects, no bad immune reactions, no hidden problems over time. Think about where devices touch you. Some sit on the skin, like a wound patch. Some touch the lining of the mouth or nose. Some meet blood, like a stent. Others go deeper into tissue or bone, like a hip implant. One more thing matters: how long they stay. Is it only a few hours, a few weeks, or months and years? These two answers, where it touches and how long it stays, guide which safety tests you need. Now, the common tests explained in everyday language. Cytotoxicity asks, do nearby cells stay alive? Picture skin cells under a bandage. Do they thrive or die off? Sensitization asks, could repeated use trigger an allergy, like a delayed rash? Irritation asks, does the material make skin or a mucosal surface turn red, sore, or swollen? Hemocompatibility asks, how does blood react? Does it clot too easily, or do red blood cells get damaged? Systemic toxicity asks, if tiny amounts enter the bloodstream, can organs like the liver or kidneys be hurt? Implantation asks, what does tissue right next to an implant look like over time? Calm and healing, or angry and inflamed? Sometimes we also look at long-term DNA trouble, genotoxicity, or fever-causing substances, pyrogenicity, depending on risk. Biocompatibility is not only about the base material, it's also about what can leak out. Titanium in a hip implant is very stable. PG hydrogels are usually gentle, but they must be clean of leftover chemicals. Polyurethane can work well, but certain types may break down and create unwanted byproducts. Always ask, what might come out of this device while it sits on skin, in the mouth, in blood, or inside bone? Two quick stories show why this matters. First, polyurethane-coated breast implants raised concerns because breakdown products looked risky. They were withdrawn from the market. Second, the Sulzer hip implant recall in 2000 was linked to a manufacturing residue that irritated tissue and led to loosening. Lesson learned: chemistry and clean manufacturing are part of safety. Here's a simple plan you can reuse. Step 1. Map contact. Where does the device touch? Skin, mucosa, blood, tissue, or bone? And for how long? Step 2. Do chemistry. List what might leach out. This is chemical characterization. Identify and measure potential chemicals under conditions that mimic real use. Step 3. Do the tox math in plain English. Estimate how much a patient might be exposed to and compare it with a tolerable level from toxicology. Step 4. Choose tests based on risk. Pick only the tests that fit your contact type and duration. Write this into a biological evaluation plan, and later complete a biological evaluation report with your results and reasoning. Pocket rules help. If a device touches intact skin for a short time, focus on cytotoxicity, irritation, and sensitization. If it communicates with blood, add hemocompatibility and consider fever-causing substances. If it's an implant or stays in the body a long time, look harder at systemic toxicity, genotoxicity, and implantation testing. Remember, materials with a strong clinical history help, but processing aids, adhesives, colorants, and residues can still matter. Keep clean processes, control additives, and document everything. After launch, keep listening to real-world feedback. Track complaints about redness, rash, pain, or unexpected reactions, and update labeling when needed. In short, biocompatibility means safe with the body. Use the contact location and time to decide tests. Do the chemistry, do the tox math, choose tests based on risk, document clearly. That is how you protect patients and keep devices on the market. 30-second short version. Biocompatibility means a device can touch your body without causing harm. To choose the right tests, answer 2 questions. Where does it touch? Skin, mucosa, blood, tissue, or bone? And how long does it stay? Hours, days, or months and years? Common tests in plain language: cytotoxicity, do cells stay alive, sensitization and irritation, rash or redness, hemocompatibility, how blood reacts, systemic toxicity, whole body effects, and implantation, local tissue response. Sometimes we add genotoxicity or pyrogenicity based on risk. It's not just the material, it's what can leak out over time. Do chemical characterization, compare likely exposure with safe limits, and then choose only the tests you need. Write your plan, run the studies, report the results, and keep watching after launch. That's biocompatibility in 1 minute. 3 quick hooks. Biocompatibility boils down to two things: where the device touches your body and how long it stays. Those answers drive the tests. If it touches skin, blood, or bone, it has to play nice. That's biocompatibility: safe materials, clean chemistry, and the right tests. It's not just the material, it's what can leak out. Do the chemistry, do the tox math, then test based on risk.